Welcome to all. In this video we will see about bacterial transformation. Bacterial transformation is important for two major purposes. One is to express recombinant protein in prokaryote system. Second one is to obtain the recombinant DNA molecules for higher organism transformation procedure. Bacterial transformation is a prerequisite. So now we will see how bacterial transformation helps in recombinant DNA technology. So to produce the recombinant DNA technology after cloning the gene of interest, the first step is transformation, the process of introduction of the DNA in the form of plasmid into a competent strain of bacteria. The competent strain means the strain which can capable to intake the foreign DNA. So once the foreign DNA plasmid enter inside, the recombinant DNA production will take place by the way of the bacteria replicate the sequence of interest. Next, this recombinant DNA plasmid also moves to the progenies of the bacterium. So the recombinant DNA plasmid will multiply tremendously. That process is called recombinant DNA amplification. This is a key step in molecular cloning to produce multiple copies of recombinant DNA molecules. So next step is we can collect the pellets of this bacteria and we can purify the recombinant DNA molecules that contains our gene of interest for transformation purpose in higher organisms or for any experimental requisite. So for this purpose the bacterial transformation is essential for the molecular biology. Now we will see the basic steps involved in the bacterial transformation. In general, the transformation in bacteria is carried out by 4 steps. The step 1 is preparation of competent cells. So already we discussed competent cells means the cells which can capable to intake the foreign DNA inside. So we have to do some pre-treatment of the bacteria E. coli cells. By two ways we can make the E. coli into competent cells. One is by chemically treat with some chemicals and call it as chemical competent cells. Otherwise we prepare the cells to be electro competent. That means under electric pulse it becomes competent to intake the foreign DNA. Second step is performing the transformation procedure. So here once we make the E. coli into competent cells, the plasmids with foreign DNA and the E. coli competent cells are mixed together and enable the plasmids to enter inside the E. coli, and this procedure is called transformation. Generally this is achieved by two methods. One is called electroporation and another method is called transformation through chemicals. The third step in the transformation is cell recovery. So here the cells recovered, that means the successful E. coli which is carrying the plasmid with the recombinant DNA inside the host is called cell recovery procedure. Next step is cell plating. So once we recover the E. coli cells carrying the plasmids, we have to culture those cells as individual colonies in the agar plate. That method is called cell plating. So here the E. coli carrying recombinant DNA are plated and this is useful for the picking up of single colony carrying the recombinant DNA. Now we will see these 4 steps one by one. The step 1 is preparation of competent cells. So to prepare the competent cells, we have to culture the purified E. coli cells without any antibiotics and culture in the plates to pick up a single colony. So from the single colony, we pick up a single colony and culture in a 1 to 3 ml of LB broth or SOS nutrient medium, and we inoculate the single colony and culture it for 37 degree for 16 hours, that means overnight, with shaking condition of 150 to 225 rpm. So the next day we will get enough colonies of pure E. coli cells. Next, to prepare the competent cells in large quantity, we have to dilute these overnight cultured cells 1:50 in the SOS medium or LB medium and culture 3 to 4 hours at 37 degrees Celsius with 150 to 225 rpm. And to confirm enough cells are multiplied, we check the turbidity and the optical density at 600 nanometer wavelength should be 0.4 to 0.6 is ideal for converting the cells into competent. Now we have to use these cells to become competent cells. We use two methods. The first method is chemically competent cells. Once we prepare these cells, we make it as a pellet, then we dilute into small quantity of water containing cationic salt such as calcium chloride or cobaltous chloride. In millimolar level, we will prepare the solution, and we have to mix the pellet with this kind of salt solutions and keep in ice for 10 to 30 minutes. Whereas in the case of electrocompetent cell preparation, we will resuspend the pellets in deionized water and keep in ice for 10 to 30 minutes. So here, once we prepare the competent cells are aliquoted into F&F tubes, approximately 50 to 100 microliter per vial, and we have to store them in the deep freezer at minus 80 degrees Celsius. To store them at minus 80 degrees Celsius, initially we place these cells in 4 degrees Celsius for 10 to 20 minutes, then keep in minus 20 degrees Celsius for 2 to 4 hours, then we transfer into minus 80 degree refrigerator. Alternatively, we can place them in the step down freezing containers which is containing ethanols, and usually they are capable to decrease 1 degree Celsius at 1 minute, and we can place these vials in such kind of boxes and directly place in minus 80 degrees Celsius. And it is advisable to add some cryoprotective agents up to 10% such as glycerol or DMS to save the bacterial cells for long time, and also we have to thaw the cells whenever we need them to perform transformation. To take the cells for transformation, we have to take them from minus 80 degrees Celsius, immediately thaw them by tap water to reach the room temperature immediately, so that the cells become active and ready to perform their role as a competitive cells. Now we will see the methods of transformation. The second step is performing transformation. Two methods are followed according to the nature of competent cells. If the cells are chemical competent, we go for chemical method of transformation. So here the cells are take away from minus 80, immediately thawed and kept in ice for 10 to 30 minutes. Usually 5 to 30 minutes. Then we have to mix the plasmids usually in 2 microliter of distilled water or 2 microliter tea buffer which contains 10 to 20 nanogram of plasmid DNA. Gently we have to mix with pipette tip and again keep in ice box for 5 to 30 minutes. After that, we take the vial and place in a water bath having 42 degrees Celsius, and just we have to expose only 30 to 45 seconds. This process is called heat shock process. So after completion of the heat shock, again keep the competent cells plasmid mix in ice and again wait for 5 to 30 minutes. The second method is called electroporation. In this method, we have to place the cells in ice for 5 to 30 minutes and add approximately 2 microliter of tea buffer or distilled water containing 10 to 20 nanogram of plasmid DNA. Then we have to gently mix with competent cells. Again keep in ice for 5 to 30 minutes. Then transfer this mix in the electroporation electrodes. After that, we have to place in the electroporation machine and apply a short electric pulse, approximately 15 kilowatts per centimeter for 1 second to 2 seconds. So once it is completed, the electroporated cells are again kept in ice for 5 to 30 minutes. The transformation process will be successfully completed. The choice of performing chemical method or electroporation method depends on the transformation efficiency of cell types and experimental goals and available resources in the lab. The third step is cell recovery. The transformed cells are mixed with 1 ml of pre-warmed SOC medium with or without the selection agent such as antibiotics. Then it was cultured in an incubated shaker at 37 degrees Celsius for 1 hour at 150 to 225 rpm. The SOC medium, which contains glucose and magnesium chloride, is recommended to maximize transformation efficiency. The fourth step is called cell plating. The cells are plated on LP agar with appropriate antibiotic, or we can go for blue-white screening if such genes are present in your plasmid backbone. So for that purpose, we need the chemicals such as XGAL and IPTG must be present in the agar plate. A sterile hockey stick or L-shaped cell spreader is commonly used to spread the cell suspension while gently rotating the plate. Thank you.